Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Process Intensification in AAV Gene Therapy. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending me a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Virica Biotech, who developed the content for this presentation. Virica is the first company to develop and commercialize viral sensitizers as a solution to the challenge of antiviral defenses in the manufacturing of viral vectors. Now it's my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Dr. Sara Wootton. She's Associate Professor at the University of Guelph. Dr. Wootton is a molecular biologist with over 20 years of experience in AAV vector engineering for a wide range of in vivo gene transfer applications. And next, I'd like to introduce you to Professor Amine Kamen. He's a professor at McGill University. Professor Kamen has expertise in the fields of bioprocesses, optimization, scale-up, viral vaccine manufacturing, viral vectors, and nanoparticles, cell culture engineering, and process analytical technologies and process control. And next, I'd like to introduce you to Dr. J.S. Diallo. He's the CEO of Virica Biotech. Dr. Diallo is a scientist with a broad expertise in biochemistry, molecular biology, cancer therapeutics, and viral immunology. Dr. Diallo is an internationally recognized expert in oncolytic viral therapy and was seminal in the discovery of viral sensitizers and its development for virus manufacturing. Now it's my pleasure to pass the mic over to Dr. Wootton. So Dr. Wootton, when you're ready, you may begin.
Great. Okay. Well, thank you very much for the introduction. I'm very excited to be here today to talk about intensification of AAV gene therapy. And today I'm going to be telling you a little bit about the biology of AAV and its use in gene therapy. Okay, so I like to think about AAV as a small but mighty virus, but it has huge potential, as you will see throughout this talk. To begin with, AAV, or adeno-associated virus, is just a small, non-enveloped single-stranded DNA virus belonging to the family Parvoviridae in the genus Dependovirus. And the reason it's in this genus Dependovirus is because on its own, it can't complete its replication cycle. So it requires a helper virus, such as adenovirus, to fulfill some of the aspects of its replication cycle. It was discovered way back in 1965 as a contaminant in an adenovirus preparation. And currently there are 12 natural serotypes of AAV that have been classified into 5 different clades, A through E, and these are found in humans and non-human primates, at least these natural serotypes that I'm describing. This virus is non-pathogenic. It hasn't been linked to any human disease. The majority of people in this audience have been exposed to AAV, and there have been no adverse events as a consequence. So wild-type AAV in its various different serotypes does infect humans, and therefore there is reasonably high seroprevalence in the human population, ranging between 50 to 80%. And again, this varies depending on the serotype. So before we get into talking about how one engineers an AAV vector, I thought we'd start by talking about the genome. So the genome, just going to get my laser pointer, shown here is small, so it's about 4.7 kb in length. It's a single-stranded piece of DNA, and on either end of the genome are these inverted terminal repeats, which are important for initiating DNA replication and also importantly for packaging. The virus only expresses 2 genes, the rep and the cap. It has these 2 open reading frames, which are expressed in this case of the rep genes from 2 different promoters, giving rise to 4 different replicase genes that are involved in replicating the genome, and then accumulating single-stranded DNA for packaging and various other functions. And then we have the cap gene. So the capsid, this cap gene encodes the capsid, which is expressed from one promoter, the P40 promoter, and there are 3 different versions of this capsid. They're all in the same reading frame. VP1 is translated from the unspliced RNA, whereas VP2 and 3 are translated from the spliced RNA. And you'll see here in this Western blot that these various capsid proteins are assembled into the virus particle in different ratios, generally speaking a 10 to 1 to 1 ratio. So with 10 particles of VP3 to 1 VP2 to 1 VP1. More recently, 2 additional reading frames were identified and their protein products, including this protein called AAP, which is an assembly accessory protein, which helps facilitate these individual subcapsid subunits back into the nucleus so that they can form virus particles within the nucleus. And the most recent reading frame identified is this membrane-associated accessory protein, which the function of which is a bit dubious at the moment. Okay, so in order to make an AAV vector, you essentially get the genome. So you remove the rep and the cap genes, leaving behind the inverted terminal repeats, as you can see here. And so this gives you about 4.5 kb to work with. You have to include your promoter of interest, the transgene, and then any other regulatory or immune modulatory elements you need, including a poly A tail. Oftentimes people will include a WPRE regulatory element as well to enhance RNA stability and protein translation. So all of this is packaged into a capsid, and that is as a single-stranded piece of DNA. Once that vector transduces a cell, the single-stranded DNA has to be converted into double-stranded DNA. So there are lots of evidence of these different versions of these AAV vectors. One that is relatively common is what we call self-complementary AAV genomes. So these are genomes where they have a slight modification or deletion in one of these inverted terminal repeats. And what that does is it doesn't allow for resolution of these ITRs. And in the end, this produces a double-stranded genome as opposed to a single-stranded DNA genome, as you can see here. The consequence of this is that the coding capacity is effectively cut in half. However, the benefit is that because this genome is now double-stranded, it bypasses one of the rate-limiting steps in AAV transduction, which is actually converting single-stranded DNA to double-stranded DNA. And so transduction can happen faster. And just this vector actually is used, or this type of vector is used in Zolgensma, which we'll talk about a little bit later. Now, there are a lot of genes that surpass the coding capacity of AAV. CFTR is a good example. So this is a very large cDNA, about 4.3 kb in length, which would not be able to fit into an AAV genome along with its promoter, poly A tail, and various other regulatory elements. And so what scientists have done is developed what we call dual vector or overlap vectors, where essentially the coding sequence is split into 2 different vectors. So this does require co-transduction with the 2 vectors. And there are various different strategies that have been developed. One of which utilizes just a region of homology between the 5' end of the gene and the 3' end of the gene. And because AAV is highly recombinogenic, homologous recombination happens rather frequently. Other methods take advantage of the fact that inverted terminal repeats also recombine into these concatemers. And so splice donor and acceptor sequences are placed on either side of the genome, and this allows for splicing out of the inverted terminal repeat and reconstitution of the gene. And then the third approach basically takes into consideration these 2 applications. In this case, the region homology is actually a region of DNA that is highly recombinogenic, that is placed in these genomes to facilitate more efficient recombination. So this is how you overcome the small coding capacity of AAV. Okay, so how do we make AAV? So basically, you require 3 plasmids. In some cases, people will utilize a 2-plasmid transfection system, but you need these 3 components. So you need a genome containing plasmids. So you have your transgene of interest with its promoter, et cetera, between these 2 ITR sequences. That gets co-transfected along with a packaging plasmid, which encodes the replicase gene, which is typically from a serotype 2 AAV, and the capsid gene. And this capsid could be from any serotype or novel serotype that you wish. So Early on, when AAV vectors were made, these HEK293 cells would be co-infected with an adenovirus to provide the helper functions. But we've moved away from using replication-competent adenoviruses to provide the helper functions because, again, you have to separate out the AAV from the ad. And so now these helper genes are just encoded within a plasmid, and that is co-transfected along with the packaging plasmid and the transgene plasmid. Sometimes when people use two plasmid systems, the rep, cap, and helper genes are all within the same plasmid. These are co-transfected into HEK293 cells because they do encode the E1A, E1B from adenovirus, which is also a required helper gene. Approximately 3 to 5 days post-transfection, virus particles are harvested, and scientists will either harvest the supernatant or the cell lysate or both, depending on where the virus particles tend to accumulate. And most often, people are using affinity chromatography to purify virus particles. But Dr. Amine Kamen will tell us more about virus purification. So that's sort of, generally speaking, how one would make AAV. Now, I mentioned that there are a lot of different serotypes, and of course, these serotypes to some extent have unique tropisms, although if you look at this panel of AAV 1 through 9, you can see some of these serotypes are quite pleiotropic, including AAV 9 and AAV 8. So there are a lot of the specificity for AAV tropism actually comes from the promoter that's utilized. But in any case, just to highlight a few of these examples, so yeah, AAV9 and AAV8 are commonly used in human clinical trials, particularly for skeletal muscle, the eye, liver, and the central nervous system. AAV6 is also used for the lung and the liver, and AAV1 is also very good at transducing skeletal muscle. So AAV2 is also used in the eye, but all of these capsids have a range of different tropisms. And that has a lot to do with the receptors that they use. So below here is a table indicating the primary receptors that these serotypes use. These are modifications like sialic acid or heparin sulfate that are found on proteins on the surface of cells. They're considered the primary receptor. And then the co-receptors include a variety of different molecules, including epidermal growth factor receptor as an example, laminin R, and more recently, a near universal AAV receptor called AAVR was identified. Not all serotypes use it, but a majority do. And this has actually been, people have capitalized on this as a way to develop affinity chromatography methods for purifying a wide range of serotypes of AAV. Okay, so the capsids I just described before are naturally occurring isolates. And I mentioned on the first slide that a majority of individuals will have been exposed to natural wild-type AAV and thus will have pre-existing immunity. And that is one of the major barriers to AAV transduction. And so a lot of people have been spending time evaluating ways to engineer AAV capsids to evade the immune response and also to try to confer some sort of tissue specificity, as well as finding ways to try to cross the blood-brain barrier to allow for systemic administration of AAV vectors rather than having to go intracranially, for example. And so there are a number of different approaches that are being utilized, including what we call directed evolution. So this involves co-infecting or transfecting with capsids from all these different serotypes that I mentioned earlier, as well as other serotypes, digesting these DNA fragments and then allowing for recombination to occur. And then you apply some sort of selection process, whether that is evasion of antibody or immunoglobulin or transduction of a particular tissue of interest. And then you can refine your search and isolate capsids that have specific phenotypes that you're after. And the same sort of approach would work for this PCR approach. So using error-prone PCR to amplify the capsids and then applying a selective pressure. There's a rational design approach where a region of the AAV2 capsid in particular has been identified that is able to accommodate inserts with a range of sizes without compromising vector packaging, particle assembly, or transduction. And so a number of peptides have been inserted into this region to facilitate AAV attachment to a specific receptor. This has been successful with AAV2, not as successful with other serotypes because, again, we still don't understand all the nuances of the capsid protein structure and coding sequence. So these insertion of these peptides may inadvertently lead to poor transduction. There are people looking at in silico design, which is looking for actually looking through genomes, looking for integrated AAVs and cloning out the capsids, so trying to isolate or resurrect ancestral AAVs. And then a much simpler way is to what I call bioprospecting. So discovering natural AAVs by using degenerate primers that pick up the capsid gene from tissue samples that you acquire from a wide range of different species. Okay, so now that we have our virus particle produced, it has to enter into a cell. And one of the interesting caveats about AAV is that it works very well in vivo, but not very well in vitro. So a lot of the transduction phenotypes that you observe in vitro don't necessarily translate in vivo. So a lot of times people will actually go right in vivo to see how their capsid is working. So nevertheless, we have an AAV here that is binding to its receptor. It's internalized by receptor-mediated endocytosis using clathrin-coated pits. And then it follows a series of low pH endosomes. And at a certain pH, there's a conformational change in the capsid, which releases a lipoprotein lipase domain in VP1, which facilitates exit of the capsid, an intact capsid, out of the lysosome or endosome into the cytoplasm. At the same time, a nuclear localization signal in VP2 is revealed, which then traffics the capsid to the nucleus, still intact. It doesn't uncoat until it gets into the nucleus. So this is a precarious step in the AAV life cycle in that a lot of these particles are inadvertently shuffled to the proteasome, they get degraded, and of course these peptides can be presented on MHC1, which is a problem with respect to activating the cytotoxic T cell response. So nevertheless, when particles make their way into the nucleus, they are uncoated, and the single-stranded DNA is converted into double-stranded DNA, and these often take the form of concatemers. And so these episomal circular concatemers are highly stable, and they reside in the nucleus essentially for the lifetime of the cell and continue to express the therapeutic transgene of interest. There is, on a very low level, integration into the genome. Again, there haven't been any incidents of cancer or insertional mutagenesis due to AAV gene therapies, but anytime DNA finds its way into the nucleus, there's always the potential for integration. So while AAV is a phenomenal gene transfer vector, there are some formidable barriers faced by the immune system. And I'm just going to touch on a few of these. So because a majority of people have been exposed to wild-type AAV, pre-existing immunity, including neutralizing antibodies, are a problem. And so if vectors are delivered systemically, this can lead to neutralization of the virus particle before it even targets a cell. There are, of course, intracellular sensing molecules like TLR9 will detect the single-stranded DNA genome. TLR2 detects the capsid. And then, as I mentioned previously, those capsids that don't make their way into the nucleus can be degraded. And then those peptides are presented on MHC class 1, which activates a cytotoxic T cell response, which can then lead to killing or clearance of vector transduced cells. And also, in some cases, when the therapeutic transgene is novel to the host, so they've never been exposed to this protein, that can also potentially be immunogenic. But there are numerous strategies that are being employed to overcome these immunological barriers, and I'll just touch on a few of these without going into any kind of detail. But in terms of overcoming strategies to overcome genome sensing, depleting the genome of CpG motifs has been shown to be beneficial and enhances transduction. Inclusion of TLR9 inhibitory sequences will dampen the immune response and also facilitate transduction. Administration of hydroxychloroquine prevents interaction between the DNA, TLR9, and cGAS, and then blocking reverse transcription from the ITRs, which do have some residual promoter activity. All of these can help suppress the innate immune response. In terms of dealing with capsid-specific immunity, primarily the neutralizing antibody response, there are different pharmacological agents that could be utilized, including rituximab, which is a B-cell depleting antibody, rapamycin, which inhibits B and T cell activation by basically making that such that they don't detect IL-2. There are enzymes that non-specifically cleave circulating IgG, so they're transiently depleted. plasmapheresis, or various structural modifications to the capsid that I alluded to on the previous slide that will delete epitopes that are antigenic, as well as directed evolution. If you're delivering the AAV vector systemically, complement is something that needs to be taken into consideration. So there are various complement inhibitors that can be pre-administered. There are antibodies that bind to C3 and C5, essentially shutting down the complement response. And then again, neutralizing antibodies would be beneficial for inhibiting the complement response. And then finally, strategies to inhibit immune cell mechanisms include proteasome inhibitors, so to prevent the peptides from being presented on MHC class 1, various capsid modifications. This is sort of an interesting idea using microRNA detargeting, so encoding microRNA sinks within the AAV genome so that if the vector finds itself in an antigen-presenting cell, such as a dendritic cell, those microRNA binding sites will bind to, for example, miR-142 that is highly expressed in dendritic cells and degrade the RNA. So there'll be very little opportunity for protein to be presented by that antigen-presenting cell. And there are various other clever methods using vipers, which are viral proteins that interfere with MHC class 1 presentation. So the list goes on, but there are a lot of people working on ways to overcome these immunological barriers. So now I want to talk a little bit about the clinical landscape of AAV. So AAV is the most widely used gene therapy vector for in vivo applications. It has a strong safety profile. There are over 200 registered clinical trials ongoing at the moment. And we actually have three gene therapies that have received regulatory approval. So the first was Glybera in 2012, and this was for a disease called lipoprotein lipase deficiency. And this utilized AAV1, and the dose used for these gene therapy trials was 1 × 10^12 vector genomes per kg at a cost of $1 million. At the time that Glybera received EMA, or European Medical Association approval, this was the most expensive drug ever made. But as you'll see, some of these this actually looks like a steal now. But one of the reasons why Glybera is no longer on the market is because of the hefty price tag. But I think our colleagues at the NRC are going to be doing something about that soon. The second AAV gene therapy to receive FDA approval was Luxturna. This was in 2017, and this is to treat an inherited retinal disease that causes blindness. In this case, AAV2 was utilized to deliver the RPE65 gene so it's a subretinal injection, and you'll see that the dose is much smaller here. This was 1.5 × 10^11 vector genomes per eye, and the price tag is lower, although not that much lower. That's $850,000 for both eyes. So you could get a deal if you just did one eye. But okay, and then Zolgensma, which is the most recently approved FDA gene therapy. And this is for a pediatric disease called spinal muscular atrophy, or SMA. This, again, is a self-complementary AAV genome delivered using an AAV9 serotype capsid. And this is delivered systemically. So the Glybera was delivered intramuscularly, and this is a systemic administration. So you can see that the doses are much higher. So we're looking at 1 × 10^14 vector genomes per kg, and the price tag for this is $2.1 million. And just for some context, so there are other gene therapies that have been approved, and more recently, Zynteglo from Bluebird. The price for that, which is an ex vivo lentiviral gene therapy, is around $2.8 million. So actually the AAV gene therapies are a little bit cheaper. But now we need to talk about dosing, because herein lies the problem. So this is a meta-analysis from 2022 looking at the average dose of AAV gene therapies based on a therapeutic area. So these are average doses. If we look at AAV gene therapy for musculoskeletal diseases, the average dose per patient was 9.1 × 10^13 vector genomes per kg. So if we assume the average person weighs 60 kilograms, that's 6.4 × 10^15 vector genomes per person, which is a lot of vector. I'm just thinking more from a production point of view, but that's also a lot of vector to be administered. If we look at some of the other therapeutic areas, including ophthalmology, you'll see that the doses are much lower, because these are being dosed based on the eye. So anytime an AAV can be delivered to a target organ, that doesn't require systemic administration, for example, the eye, the brain, potentially the lung, the doses are going to be lower and thus more affordable. So yeah, these are the range of doses for therapeutic areas. And then in terms of what's happening in these clinical trials in terms of serotype usage, here's a pie graph showing that the major serotype being used currently is AAV2. And I think a lot of that has to do with the fact that a lot of the clinical trials are aimed at diseases of the eye, and AAV2 is very good at transducing the eye, and also that AAV2 was the first gene therapy vector or first AAV vector to be developed and moved through the clinical translation pipeline. But you'll see that AAV8 and 9 and 1 are also taking up reasonably large slices of the pie, and this is most likely going to change over time in the next few years, and AAV6 is a good contender that will probably take up more of the pie. But this is just sort of a snapshot of what's happening recently or in the last few years. In terms of therapeutic targets, so the major targets include the brain, the muscle, the liver, and the eye. A notable organ that's missing from this pie chart is the lung, in part because we didn't really have any AAV vectors that were efficient at targeting the lung, but now we have a number of vectors that are lung tropic and are very good at transducing the lung, including one that we use in our lab. And so I have a feeling that lung might actually make its way onto this pie chart soon. And then here's where the AAV gene therapy trials are in terms of with respect to phase. They're through all, majority are phase 1, phase 2, but there are some therapies that are in phase 3, and as you'll see in the next couple of slides, are waiting for FDA approval. So it's interesting to see all these gene therapies and all the different trials that are ongoing, but are they efficacious? So again, in this meta-analysis that was published in 2022, this is a chart that shows by therapeutic area whether or not these clinical trials, in this case they sampled 98 clinical trials, whether or not they met their efficacy endpoints as well as safety endpoints. And one thing I was pleasantly surprised to see is that if you look at the blue bars, a majority of these therapeutics, in these therapeutic areas, a majority of these AAV treatments are achieving therapeutic efficacy, which is very exciting. Okay, so this slide is very busy, but it's not meant for you to read, but more just to give you a snapshot of the various gene therapy products that are currently awaiting approval from the FDA. And I've gone through and highlighted the various AAV gene therapies that are awaiting approval. And just for some perspective, so of the 69 therapies that are awaiting approval, 26 are AAV gene therapies, so that represents about 38% of all of the gene therapy products awaiting approval. And so this includes gene therapies such as ex vivo lentiviral vectors, mRNA, DNA, so AAV is a dominant player in the gene therapy field. Okay, so this is my last slide. So I thought I would just end by summarizing some of the key advantages and of course some limitations with AAV before I pass the virtual platform over to Dr. Amine Kamen. So some of the clear advantages of AAV include the fact that it is poorly immunogenic, in particular, like with respect to other AAV gene or with other gene therapy vectors. And this, the fact that it is poorly immunogenic is one of the reasons why we have sustained transgene expression from this vector. It's non-pathogenic. It is able to transduce both dividing and non-dividing cells, although transduction of dividing cells, particularly rapidly dividing cells, will ultimately lead to a dilution of the vector and a waning of the therapeutic transgene expression. The virus or the vector persists episomally. It doesn't typically integrate, and so this persistent episomal presence leads to this constitutive transgene expression, and this is both robust and sustained, and there are individuals who have been administered AAV who are expressing therapeutic transgenes beyond 10 years. So it's highly effective, and it is the most efficient in vivo gene transfer vector. In terms of its limitations, I touched on this earlier. It does have a limited coding capacity of about 4.7 kb, excluding certain therapeutic genes such as the CFTR gene. However, there are ways to potentially overcome this packaging capacity. Pre-existing immunity to the wild-type serotypes is an issue. There are various chemical methods that can be used to deal with this pre-existing immunity, or capsid evolution is happening to try and select capsid variants that no longer are neutralized by the antibodies prevalent in the hosts. As I mentioned in the previous slide, very large doses of AAV are needed for efficient transduction, especially if the therapy needs to be delivered systemically to the entire skeletal muscle system, for example. And then another thing to consider is that a number of these gene therapies are being utilized in pediatric populations. So at some point, re-administration of this vector is going to be necessary. And so we need to figure out how to dampen the initial immune response to the gene therapy so that we can eventually re-administer years down the line when and if that becomes necessary. And then I thought I would just again leave off with some discussions about doses used in clinical trials, just to set the stage for Amine's talk. So if we look at, again, this is from a meta-analysis, this is the range of doses that are used for targeted administration in human clinical trials. And so what I mean by that is, again, targeting an organ such as the eye or the brain. And you can see that, you know, these doses seem reasonable in the 10 to the 10, 10 to the 11 vector genomes, you know, upwards of 10 to the 14. However, when we look at systemically administered AAV, these doses start to climb dramatically, even upwards of 10 to the 16 vector genomes. And so I guess my final comment is that manufacturing capacity improvements are desperately needed in order to increase the yield of vectors so that we can get these very promising gene therapies into the clinic and ultimately commercialized so that we can save lives. And I have some references, but at this point, I'm going to turn the platform over to Dr. Amine Kamen. Thank you.
Thank you, Sara, for quite complete introduction actually on the AAV. This will definitely facilitate my presentation, and I will try to catch up on the time. So the introduction here is, or the first slide is about mentioning actually the National Research Council of Canada, where I did a lot of work actually prior to joining McGill University, and some of the work has been also done with a McGill spin-off actually called Virica Bio. Next slide, please. So I will go briefly about, I think Sara did the introduction. What are the two dominant technology, transient transfection and insect cell technology? And I will touch on two slides on the downstream processing and analytical, and that I will go with the perspective, actually, what's currently moving forward toward intensification, because this is the topic of this webinar. Next slide, please. So just for the sake of completeness, actually, there is different technologies that have been used essentially to deliver what Sara had described, the key component, the gene of interest, the rep and cap, and the helper function by transient transfection. Other means to deliver this component was through the helper simplex virus. This technology was at some point used. Baculovirus system I will describe briefly. And there has been in the past already producer cell line based on the HeLa cells using the adenovirus, we should say here packaging cell line. Next slide, please. So I think this, Sara went extensively on the topic, actually, with regard to the dosing. This is another way just to present it, that some diseases are more easy when it's local administration versus systemic administration, showing the number of patients that you can treat with one single production at 10 to the 16, say 100 liter scale. Next slide. So among the technology, 293 cells have been used. So our contribution at the early time was to make the process in suspension. And this was using the cell line that has been adapted and deposited as a master cell bank. Next slide. And the technology transient transfection has been developed to operate in suspension. Okay, everyone is familiar with transient transfection with adherent cells. So the move and the scale up was to adapt this transient transfection in suspension using bioreactor by the three component rep cap plasmid, cDNA, the gene of interest, and the helper plasmid, as described by Sara, using PI, polyethyleneamine, as a vehicle for transient transfection. Thereafter, you harvest the cells. Next slide will show the timing of the operation. This can be operated up to the 60-liter scale. So here we are talking about intensification in terms of scale to generate large quantity of material. We start from 0.25 million cells per ml, let them grow to 1 million cells per ml. DNI, PI have been optimized the ratio, incubate 15 minutes, and inoculate in the bioreactor where cells have been grown, and the harvest, depending on the production, might be 3 to 6 days later. And this can be repeatedly operated at 60-liter scale bioreactor. That's work coming from the NRC. Next slide, please. So the same process has been scaled up to the 500-liter scale. So this is showing real operation actually and demonstration of the feasibility of the technology. Although complex with 3 plasmid, it is definitely achievable to meet large need of AAV. Next slide. Other technology actually that was used in a number of products that has been licensed is the baculovirus technology. And in short, in fact, you can use the key rep cap and the gene of interest coded in the baculovirus and infecting the cells in this case. So you see the value actually, infection is far more effective than transfection. Other constructs have been made using just 2 baculovirus or eventually 1 single baculovirus holding different component and coding eventually the sequence on the cell line, having a packaging cell line. I will briefly describe this technology. Next slide. So this is building on major achievement that have been done with the baculovirus system. In fact, that goes back to 2010 with this 3 baculovirus system, infecting the cells and letting the cells grow. My time of infection is time 0, as indicated here. You see that we can achieve a cell density up to 40 million cells per ml if we don't infect. If we infect and we fat batch, adding certain nutrient, as shown here, 3%, 5%, 7% from the total volume of the bioreactor, we can produce at cell densities that are in the range of 10 million cells per ml. So it's basing the further work on this well-developed technology on the baculovirus in insect cell that we were able to intensify the process. Next slide. So, and this process has been demonstrated as robust to be transferred to GMP manufacturing site. This showing just as a demonstration that this can be repeated at 45-liter scale. Shown here the different step in the manufacturing from the first lysate collection, filtered, ultrafiltration, and affinity column eluate, reaching beyond 10 to the 14 VGs. Next slide. So the technology that was put forward, and this is toward the packaging cell line. In fact, this work is well described, and I will not go in detail. Minimized, in fact, the baculovirus to a single baculovirus, and using a packaging cell line where the rep and the cap function were coded. This work was done in collaboration with Zoritkin from Florida University. Next slide. And this enabled combining, in fact, the 2 technologies, the high cell density with the baculovirus system, as well as the packaging cell line, to shortening the demonstration here by using fat batch operation to multiply the yield by 45 to 60 times. Obviously, this is an extensive summary of extensive work that has been done. This summarizes, in fact, a PhD work over a couple of years. Next slide. And if we take the different system and try to compare them, so at the very first, I am showing that with this advanced system, packaging cell line, baculovirus, single baculovirus, you can produce actually a dose of 10 to the 16 in 25 to 50 liter scale. At the other limit, if you use the transient transfection, to get this level of production, you need to consider a 1,000 liter scale. And in between, these are the scale that might be required by less advanced technology using the insect cells and the baculovirus system for production. So this gives an idea of where we stand in terms, in fact, of scale up and ability to produce preclinical and clinical material. Next slide. Another, and I will just touch on this, it hasn't been alluded to, but one of the critical limitations in any of the manufacturing process is the inability to make totally functional AAV particle, and we end independently of the technology by generating a lot of empty capsids. So there is a limitation. This is showing what you get by analytical ultracentrifuge as a distribution. For years, in fact, going back to the 2000, this has been ignored actually, but currently this is more and more a requirement to get a separation. So what's the basic of the separation of this very, very tiny difference between empty and full AAV particles that are totally functional? Ultracentrifugation has been used, but anyone might guess the limitation and the capital investment that is required. Next slide. So for this purpose, we moved to a chromatography after the immunoaffinity that has been alluded to by Sara. There is a second step using anion exchange chromatography to enable, in fact, separation, empty and full capsid. Next slide. And this is a method fully demonstrated with the AAV5 to enrich, in fact, the production lots in terms of full vector particles that are totally functional. Next slide. I'm going fast on this because all this has been published. So this has been demonstrated also that this method can work very effectively with a different serotype. Next slide. So we are moving from the ultracentrifugation, that is the only method currently possible actually to separate the empty versus full particle, to chromatographic approach, and this has been demonstrated as working effectively. The same guiding principle with ion exchange chromatography can be used at the analytical level and can, next slide, eventually replace the analytical ultracentrifuge, that is an analytical tool based on ultracentrifugation once again, by an operation using an HPLC that is more accessible in different labs. So this shows that we can reach significant yield actually using the insect cell technology. In the downstream process, this is considered also contributing to the intensification of the process. We can use chromatographic method based on ion exchange, and we can use more accessible tools such as HPLC, in fact, to quantify in the preparation the ratio empty versus full capsid. Next slide. And these are my conclusion actually about the different process that I have. All of the HK293 are a good system to produce by transient transfection. the approach with the insect cell generated significant result, and in fact, many of the products that have been licensed were using insect cell technology. We believe, I strongly believe, and this is what might be indicated by the insect cell technology, that producing and packaging cell line are definitely the way to go for further intensification of the process. Why? This has been demonstrated by the dosing that is required for systemic delivery, especially if we think about treatment of large populations such as sickle cell treatment. We are beyond the orphan disease, and we need certainly to continue developing process analytical technologies to monitor the process and to enable, in fact, further optimization of the process. Last slide, I believe. So this involves a number of people, and I will ask the next speaker to take the stage.
Thank you very much, Professor Kamen. But before we have Dr. Diallo take the stage, I'm going to launch our first poll question. Is that okay, Dr. Diallo? Okay, I'm going to launch it now. Okay, here we go. Here's our first poll question, audience members. We'll give you 40 seconds to complete the simple poll question. Your participation is strongly encouraged, as it will help us to get the full numbers here for representing our audience. Now, this is done in real time, so please go ahead and cast your vote. I'll give you a few more seconds, and then I'll share the results with everyone, and then we'll get to Dr. Diallo. Okay, that looks like it's the majority of our audience. Thank you so much. Now I'm going to close the polls. Oh, hold on. I'll give you a few more seconds. Remember, the question that is, what is the primary technology you are using to improve your yield? Is it cell culture media and transfixion reagents? Is it novel bioreactors, plasma design, small molecule additives, or is it other? Okay, that looks like it's everyone. I'm now going to close the polls, and let's take a look at the results. Okay, Dr. Diallo, here's the results. We have in our audience, 59% said cell culture media, while 7% said novel bioreactors. We have only 9% of our audience that said plasma design, while 20% said small molecule additives, and 5% just said others. So thank you, everyone, for participating in our first poll question. Now over to you, Dr. Diallo.
Thank you, Sonia. So yeah, it's a pleasure to be here today, and I really like to thank everybody for their participation in this webinar on behalf of Virica. Thanks to my colleagues Sara and Amine. I think they presented a very holistic vision of AAV biology and process intensification technologies. What I'm going to focus on today is really the role of antiviral defenses in AAV gene therapy manufacturing, and how they can be involved and manipulated in the context of process intensification strategy. Next slide, please. So some of the key strategies that are typically used in process intensification include cell culture media optimization, cell line and vector design enhancement, as well as bioreactor design. So in the next slide, I'm going to review a few considerations around each of these technologies. So cell culture media is one of the key approaches that is used to optimize viral vector production. And the trend these days is really to move towards completely chemically defined and serum and animal-free media. These growth media typically aim to improve cell density, improve cell growth, and they're typically involving many components, typically between 40 to 70 components. So there's a lot of opportunities and a lot of work in optimizing these cell culture media. And typically this is done in a platform-specific fashion, either specifically focusing on a cell line, for example, HEC293, or a pairing of cell line and virus. Now, cell lines and vector design are increasingly being seen in the vector manufacturing space, and sometimes the objective is twofold. For example, in the case of AAV, obviously mosaic capsids can be designed that not only improve actual therapeutic potential, as touched on by Sara, but also in some cases can actually improve viral vector manufacturing. Now, Amine touches on this as well. Clearly, there's a trend towards stable producer cell lines using technologies like RNAi, CRISPR, and a variety of others. We heard about the baculovirus system as well. Now, these can be really good strategies when you reach commercial stage in particular, but indeed they are quite expensive and time intensive. I think I heard Amine say it can take an entire PhD thesis to get this, something like that optimized. I mean, it's definitely worth your while. But once you're down that road, you have limited flexibility, and you're kind of locked into a system. And if you start off with plasmid-based production, this is definitely a significant change to your process. On the other end is bioreactor design. The current trend, as Amine mentioned, is to move away from adherent cultures and into suspension, which obviously allows for greater scalability and also increased density. But still, there is hope for adherent cultures as well, using novel platforms like, for example, macrocarrier-based bioreactors, such as the iCELLis bioreactor, for example. Now, of course, these novel bioreactors are inherently a change to the process once you move from one to the other. And obviously, training can be quite substantial to learn some of these increasingly complex instruments. But the advantage there is a potential reduction in footprint and scalability. Next slide, please. So each of these strategies can be used either alone or in combination to improve and basically intensify our viral vector manufacturing processes. But one of the things that they don't really address, and I think this is something that's an unappreciated hindrance in manufacturing, and this was a little bit alluded to by Sara at the beginning, is really these antiviral defenses that actually not only play a role in the context of the gene therapy itself when you're trying to administer it to the patient, but can also play a role at the level of manufacturing and actually hinder both vector quantity and quality. Next slide, please. So before I dive into that, it's important to understand what these cellular antiviral defenses are. So all cells have a variety of genes, hundreds of genes that are actually devoted to protect themselves against viral vectors and viral pathogens. And so they're equipped with a wide range of pattern recognition receptors that detect foreign RNA and DNA, things like RIG-I, STING, toll-like receptors. These are some common names that you might have heard of. And ultimately, these receptors aim to essentially transduce a signal that's going to lead to the upregulation of genes that will have direct antiviral effects. And so these are products that will actually degrade viral RNA, viral DNA, and viral proteins, all again to prevent viral spread in the case of infections. And this is something that's evolved over millennia. Next slide, please. So the antiviral systems are quite complex, and we tried to simplify how they work here by producing a little bit of a video that Sonia is going to share, and I'll walk you. So before I move on, I think there's a poll here, Sonia.
Yes, we'll do one quick poll question. I'll launch it right now because I know we're getting close to time. There you go, audience members. Please go ahead and participate by clicking on any of the below. What platform do you primarily use for your AAV production? Is it commercially available HEC-293s, proprietary in-house HEC-293s, or insect cells, inducible producer cells, or other? Please go ahead and make your selection. Perfect. Thank you so much, everyone. Now I'm going to close the polls. Okay, here are the results, Dr. Diallo. We have in our audience 69% saying commercially available HEC-293s, 25% said proprietary in-house HEC-293s, while 3% said insect cells, 0% said inducible producer cells, and 3% said others. So thank you everyone for participating in our final poll. Now back to you, Dr. Diallo.
Thank you, Sonia. Next slide, please. So what about antiviral defenses in the context of AAV manufacturing? So AAV actually has a wide range of places in the cell where it actually activates antiviral defense. So for example, on entry, AAV particles, and Sara touched on this already, can activate TLR2 and 9. AAV genomes can be detected by DNA sensors in the cell, such as, but not limited to, C-gas, which is an upstream activator of STING. AAV RNAs can also trigger RIG-I and other RNA sensor. And just the fact of producing AAV at high levels in a cell can actually produce stresses that are going to trigger mitochondrial activated antiviral pathways. So there's a lot of avenues for your AAV in production to actually trigger antiviral defenses at different stages of the production process. Next slide, please. So as validated by the poll, most people actually producing their viral vectors in HEC-293. And it's important to understand that HEC-293, even though they have some defects, they have robust capabilities of producing and mounting an antiviral response towards viral vector. For example, they can produce and respond to interferon that actively work to blunt viral production. They express DNA sensors like STING that transduce signals from things like C-gas and many other upstream receptors. They detect RNA via RIG-I and transduce the signal by IRF3, producing type 1 interferons, as well as the underappreciated type 3 interferon, which are also quite antiviral in nature. And altogether, HEC-293s also trigger antiviral defenses through other less appreciated mechanisms involving RNA Pol 3 and RIG-I in response to DNA rich in AT sequences. Next slide, please. So ultimately, I think it's important to appreciate that these things are happening in your AAV manufacturing process as you're producing your AAV vectors, particularly in HEC-293. So what can you do to actually antagonize those defenses? Well, the technologies that are at your disposal right now are quite limited, but essentially consist of RNAi or knockouts. And so RNAi, of course, is a transient reduction in expression of genes. Obviously, you have to do that in a very targeted way. Sometimes it's incomplete. You need to transfect in order for this to work. So it can be complicated and costly, not to mention the kinetics of knockdown. Now, you can knock out your antiviral genes by homologous recombination or CRISPR, and obviously those are long-term effects and permanent. Now, obviously, genetic stability of those systems is always a concern. And importantly, I think underappreciated is that some of the targets that are involved in antiviral defenses actually play a dual role in cell survival. And so knocking those out actually doesn't work because it actually reduces cell viability ultimately. Next slide, please. So Virica's solution to this particular problem is really the viral sensitizer platform. These are small molecules that were initially identified by high throughput screening. It's a collection of over 100 molecules that we uncovered over the years through a variety of activities, including also medicinal chemistry and mechanism of action studies. Ultimately, these compounds actually work by blocking the cellular antiviral defenses of cells in a variety of different ways in order to minimize the impact of those antiviral defenses. And because they have diverse mechanism of actions, they can be used individually or in combination. They can provide additive or synergistic effects. And they can be tailored to individual applications. So they're not really a one-trick pony. It can work in any viral vector producer cell combination. And their flexibility is really the advantage here, and their transient nature and ease of removal in the final process also a really key feature. And so these can be used to improve your manufacturing efficiency, not only improving yield, but potentially improving viral vector quality as well. Next slide, please. So I'm just going to leave you with one example here. This was a collaboration where we use viral sensitizers to help the production of AAV2 in suspension HEC-293, in this case using an Amber 15 microbioreactor system. The graphs at the top are really to show that the viral sensitizers are not really significantly affecting viral growth, lactate, or glucose. And ultimately, the use of these small molecules can actually significantly boost viral vector yield, even in these optimized processes. So we're able to achieve a 4x improvement in yield. It's important to mention that as I alluded to previously, some of the antiviral targets do play a role in cell survival. And so when you push the dosing too far, it can actually lead to reduction in cell health. But the ideal is to find that dose that basically hits the target and allows for optimal viral vector production without minimizing or compromising cell viability. So with that, I'd like to thank you for your attention. And again, I would like to thank you on behalf of Virica for attending the webinar and inviting you to reach out to us on LinkedIn and gain access to that video that I premiered a little bit earlier. And again, would like to thank my colleagues for participating in this webinar. Thank you very much.
Wow, thank you so much, Dr. Diallo. Thank you very much, Dr. Wootton and Professor Amine. We have run out of time, but we're going to try to squeeze in at least one question here. So if I can just get our speakers to put on their webcams. And it looks like this question here is for Professor Kamen. So this audience member is asking, what are some strategies that can be used to improve full to empty ratio? Professor Kamen?
Open my microphone. So
yes, there you go.
It disappeared on my screen. So what's the question? Sorry.
Oh, yes, certainly. The question is, what are some strategies that can be used to improve full to empty ratio?
Oh, so obviously I went fast actually, but I mentioned that the direct strategy is ultracentrifugation. Actually, this is what has been used for the industry that can invest at this scale actually. However, I mean, ion exchange chromatography, the method that we developed, and this is published, enable in fact enrichment up to 90% full versus empty. Okay, so there is hope that we can eventually use ion exchange chromatography to make that separation between the full and empty.
Okay, thank you.
And maybe I can add to that.
Yes, please, Dr. Diallo.
You know, again, with respect to antiviral defenses, we know that those can impact full to empty viral ratios already at the upstream. So I think it's a combination of different strategies that will be key to remove the 90% of empty capsids that you clearly see in these productions. So yes, thank you.
Dr. Diallo,
Yeah,
oh yes, yes, go ahead, Dr. Wootton.
Do we know if viral sensitizers are able to increase the ratio of full to empty?
Yes, we've seen that in some partnerships. Yes, absolutely, and it makes sense logically based on the mechanism of action of these molecules.
Okay. And Dr. Diallo, when we were playing the video, for some reason the audio wasn't coming through. Can you give us a little, a few seconds of what we were, the audience was seeing there?
Oh, was I on mute this whole time? Was that?
No, just a little part of the video. Just a little part of the video.
Yeah. So again, the video was really underscoring, the temporal nature of these antiviral defenses and the different aspects that come into play at different times. When you're transfecting with plasmid, you're triggering different things than when the virus is being produced, and there's really a kinetic change over time. And I've actually seen some interesting studies coming from Pfizer at the last ASGCT, really showing the temporal nature and the antiviral response is actually happening in these 50-liter bioreactor scales. So I think it's an important thing that people haven't really focused on too much in the last while, but it's becoming very clear that these things happen. And the more your process is intensified, the more closely your cells are packed, the more dense they are, the more these are going to be important factors in your process.
Okay, thank you for that recap. All right, well, we're going over a little bit here, but I think it was very worthwhile that we got at least one question in. There was a bunch of questions that came in from our audience, but unfortunately we don't have time to answer them. So I just want to say thank you very much to our audience members for sending them through. Now, if we could not attend to all your questions here, I just want to give you an email address that you can utilize, and the team at Virica will follow up with you after this presentation with any of your questions that weren't answered. And if you have any inquiries, please go ahead and utilize the email address that's on your screen. So thank you very much, everyone, for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event on this page, and you can also share this link with your colleagues once they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers, Dr. Wootton, Professor Amine, and Dr. Diallo for that very engaging presentation and for the conversation. We all appreciate that. Okay, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Thank you for your time. Bye.